Um, Hi, my name is Jamie Felton. I am a pediatric endocrinologist and physician scientist here at the IU School of Medicine. In my clinical time, I see lots of patients with type 1 diabetes, and as a physician scientist, I study the immune pathogenesis and really the immune drivers of type 1 diabetes development. And so for the next few minutes, I'm excited to talk to you about betting on the B team, or more specifically, a case for targeting B cells or B lymphocytes in the progression of type 1 diabetes. And before I do that, I wanted to tell you a little bit about how I got to the Diabetes Center and to be doing the work that I do. Um, I actually am not from Indianapolis. I do not have family here, so I am a transplant, and I was born in outside of Denver, Colorado. That's where I grew up. I went to college in North Carolina at Davidson. You may have heard of Steph Curry. He was in college at the same time I was. And then because I was a French major there, my real introduction to immunology and potentially work you could do as a physician scientist was really when I worked as a research technician in Jerry Palmer's lab at the University of Washington. So I moved to Washington and lived there for a couple years before I went to medical school in San Antonio, and then back to North Carolina for a pediatric residency, and then did my pediatric endocrinology fellowship at Vanderbilt in Nashville. And there I worked with my mentor, Tom Thomas, where I looked more specifically at the role that B cells play in type 1 diabetes. And so basically, I'd lived in every region of the country except the Midwest. And so it made perfect sense to, about 5 years ago, come and really start my career at the Indiana University School of Medicine, in part to work with the amazing Diabetes Center here. I had heard members of the Diabetes Center from Indiana speak at different conferences multiple times before I ever came here myself, and was very, very excited to be able to come now and call them colleagues. They're excellent scientists and also very fun people, and so we have a really, really great group. And I'm excited to get to share some of the work that I'm doing alongside all of them. So when I think about the work, and when I talk about the work that I do with type 1 diabetes, it really comes from a pretty personal place. This is a picture of my immediate family plus spouses. My immediate family growing up, my parents, and then myself and three younger siblings. And so of the six of us, 50% of the people in that group have type 1 diabetes. That's myself. My dad was diagnosed in his late 20s. My brother was diagnosed when he was a teenager. And so understanding type 1 diabetes and really the goal to prevent or delay it really hits close to home. I was actually diagnosed as a participant in a research trial. And so again, type 1 diabetes research, prevention trials also hit fairly close to home. Because my dad had type 1 diabetes when I was growing up outside of Denver in the early 90s, the Barbara Davis Center is located there, which is another fabulous diabetes center. And they were running a trial called the Diabetes Prevention Trial Type 1, where they were screening relatives of people with type 1 diabetes for autoantibodies. And if they were positive for those islet autoantibodies, enrolling them in a trial to see if a little bit of insulin could delay the onset of type 1 diabetes. So this is the postcard that we got in the mail that said I was antibody positive and really precipitated my parents to discuss with me and with our doctors the decision to try to join that trial. And so really for as long as I've been thinking about type 1 diabetes as it pertains to me specifically, it has been in this context of what can we do to prevent it. I ultimately was unable to get randomized into the trial before I was diagnosed. And overall, the trial, which was published in the New England Journal of Medicine in 2002, showed that what was tried actually didn't work. And so I think what's interesting is that this is a review that came out around the same time, titled DPT-1 Trial, a Negative Result with Lessons for Future Type 1 Diabetes Prevention. And it goes through several key points made back in 2002 that I think really guide and have motivated a lot of my work and our Diabetes Center work today. And so some of those lessons are, number one, large preventative trials are feasible, which means, as a result, other preventative approaches can be tested. The natural history of type 1 diabetes has been elucidated. We can identify those people prior to disease onset. And there does look like strict follow-up allows for earlier diagnosis, and that earlier diagnosis has long and short-term benefits. And so taken together, what this says is we can do prevention trials, we can identify people for those trials, and even if the trial fails, that earlier identification of disease has benefit. And so my goal as I think about why I got into type 1 diabetes research was really to understand the autoimmune process that happens before diagnosis so that we can identify additional targets to test in other prevention trials. I want to figure out what we need to test next. So for the next few minutes, I'm going to talk about the role that B cells play in type 1 diabetes. We will review roles for B cells in type 1 diabetes development and progression. We'll consider how B cell metabolism might be leveraged for immune modulation, and then explore some evidence for targeting B cells in early stage type 1 diabetes progression. So we've known since the early 1980s that type 1 diabetes is a chronic autoimmune disease. And that came after the publication of George Eisenbarth's paper in the New England Journal of Medicine. And preceding that had been these observations in the Lancet that individuals who had multiple endocrine deficiencies, people that we knew based on the course of other disease patterns were going to develop type 1 diabetes, people were able to identify pancreatic islet cells or antibodies to pancreatic islet cells in those individuals long before diagnosis. And so that led to looking for those antibodies in individuals who may be at risk for type 1 diabetes. And the model that came from this paper, we have beta cell mass on the y-axis and age on the x-axis. And so you can see that individuals with genetic predisposition after some sort of precipitating event develop overt immunological abnormalities. And primarily the abnormalities described were the presence of autoantibodies. Importantly, at that time, insulin release is normal. And as this immune response is progressing, you have gradual decline in beta cell mass until you reach a threshold point where you have overt diabetes and you need to start insulin. And so we know from several mouse studies, several very important mouse studies, that B cells play a critical role in that progression that happens as beta cell mass is declining, as the immune response is developing, and that path towards type 1 diabetes. We know that they function primarily, and this is through mouse studies, as antigen-presenting cells. And we also know that they infiltrate the islets. They are also cells that are invading the pancreas, the islets, and where those beta cells that are making insulin live. We see T and B cells in that infiltrate, which is what's shown here. We also know that those T and B cells are forming structures that allow them to talk to each other. So they're infiltrating the islet and they're doing something there. Like I said, a lot of this work came from mouse data. And so some important work published almost 10 years ago now that looked at B cells in human data found that if you looked at the pancreas of individuals who were diagnosed with diabetes at a young age compared to those who were diagnosed later, one of the main differences and one of the only differences was that the ratio of B cells to T cells was much, much higher in individuals diagnosed at a younger age. At the same time, when they looked at the frequency of islets that were still standing positive for insulin, they found that in that same age group, that frequency was much less. And so that led to the conclusion that this B cell infiltration characterizes a more aggressive form of insulitis that seems to be seen predominantly in individuals who were diagnosed at younger ages. And so this is also evidence that B cells are playing some sort of role in human disease as well. And so knowing that B cells play an important role, there was a clinical trial also about 10 years ago that looked at what happened when you depleted all B cells. So it was one treatment with an antibody called rituximab. All B cells were depleted, and then C-peptide was followed as a measure of the person's ability to continue to make insulin. And what they found was that at 12 months after treatment, there was a statistically significant increase or preservation of C-peptide in individuals treated with rituximab versus placebo. But that effect really disappeared by 24 months, and there really was no difference. And so the fact that we do see a change and that this was depletion of all antibodies really suggests that there is a role that B cells are playing, and it's not completely understood. One of the downsides to depleting all B cells, in addition to introducing some transient immune deficiency, is that there may be B cells that have protective effects that are also being depleted. And so could that be something that is coloring this picture and leading to the results that we see? And so what would be really fantastic would be to be able to target the cells that are causing the damage. And we call those antigen-specific B cells. And so one of the main barriers to studying those antigen-specific B cells is that they're very, very rare. And so you can see that here. These are B cells from the NOD mouse model. So this is a mouse that is going to develop diabetes spontaneously. And what you can see is that in a wild type mouse, the frequency in that upper right-hand corner where the percentage is, that frequency of anti-insulin B cells, so B cells that are going to identify insulin specifically, thus they're antigen specific, the frequency is less than 1%. However, if you skew the repertoire by inserting a heavy chain for every B cell receptor that is specific for insulin, allow it to recombine with an endogenous light chain, as a result, you increase that frequency to 1 to 3%. At the same time, if you use a heavy chain that cannot recognize insulin, you virtually eliminate anti-insulin B cells. And so this was a model that was developed by my mentor when I was in fellowship. And we used it to look at disease progression and also really parse out what is happening with those anti-insulin or antigen-specific B cells. And this model was the model that allowed us to test that. And what we found is that diabetes development was accelerated in these mice. So really just skewing that frequency of B cells, increasing that to 1 to 3%, we see accelerated disease progression in the transgenic compared to the non-transgenic mice. And so this again is an indication that those B cells, and specifically those insulin-specific B cells, are important drivers of disease progression. So we wanted to know a little bit more about what those B cells were doing, what they looked like, and how they may be playing these roles. And so when we look to broadly characterize them, I'm going to show you 2 slides and with some major take-home points here. But what we found was that overall, these anti-insulin B cells were found in 2 specific compartments or 2 specific subsets of B cell development. So as B cells develop, they go through different maturation stages, and as they progress to become the antibody secreting cell or the memory B cell, they go through different stages. And so what we found was that these anti-insulin B cells accumulated in what we call the late transitional and marginal zone stages or subsets. And at the same time, those anti-insulin B cells were enhanced for IL-10 production. And so if we looked at B cells that were positive for insulin compared to negative for insulin, we see that IL-10 production is increased. Classically, IL-10 is considered a suppressive cytokine or something that is made to suppress an immune response. IL-10 is a cytokine that's made by regulatory T cells. And so this is something that should not theoretically drive autoimmune disease. And so it's possible that if with B cell depletion, with broad B cell depletion, we're depleting anti-insulin B cells, we may also be depleting cells that could be making potentially suppressive cytokines. But we thought it was really interesting because what we see is that subsets enriched for IL-10 production contain these high numbers of autoreactive B cells. And so you have a cluster of pathogenic B cells that are making cytokines, providing help to attack the pancreas that accumulate in these subsets and that we identified in our mouse model. They were driving diabetes progression. At the same time, those cells are also capable of making a suppressive cytokine or IL-10. And so in the same subsets you see that are enriched for IL-10 production, you see similar subsets in both of those. And so that overlap really suggests that there's a subtle balance in the signals that control B cell fate. And so that led us to think about what are some of those signals that could control that balance. And if we could control that balance, can we skew that towards more of a protective rather than a pathogenic phenotype in those B cells? And so one of the things that could play a role is transcription factor HIF1 alpha. It was reported in 2018 that HIF1 alpha was a critical transcription factor for IL-10 producing cells in or IL-10 producing B cells in autoimmune disease. And so this was not a diabetes specific model. But what they found here was that HIF1 alpha mediated cellular metabolic reprogramming, or it does mediate cellular metabolic reprogramming that allows adaptation to hypoxia. And so in response to hypoxia or changes in the environment, HIF1 alpha is going to change how cells are using energy. We know that hypoxia is a very common feature of inflammatory microenvironments, and insulitis is no exception. And we also know that autoreactive B cells, because they accumulate in those late transitional and marginal zone subsets, are uniquely sensitive to environmental stimuli. And so all of that together led to the question, can autoreactive B cells be skewed toward a regulatory phenotype? We first wanted to see is HIF1 alpha even detected within the insulitis lesions within the eyelid? Because if there was no HIF1 alpha, and HIF1 alpha is important for regulatory B cells, it would make sense that you don't see any regulatory B cells within that eyelid infiltrate. But interesting, what we found was that HIF1 alpha was very much present within the lymphocytic infiltrate. And so that's what you see here. The yellow dots represent staining for HIF1 alpha, and they're found within the B anti-cell infiltrate. The B cells are in red. We were unable to simultaneously stain for T cells as well, but as you can see, we see HIF1 alpha expression in B anti cells, and importantly, we see it in the eyelid. So it's not that it's not there. And so that finding led to the hypothesis that in the setting of eyelid inflammation, dysregulated cellular metabolic reprogramming permits autoreactive B cell expansion. And so to address this, we started with two questions. One, what characterizes B cell metabolic phenotypes? And then two, what is the relationship between B cell metabolism and HIF1 alpha? Okay. To look specifically at the B cell metabolic phenotypes. And so this meaning how the cell is using its energy. What is the primary form of energy? How is it using glucose, and how are the mitochondria responding to changes? We used two flow cytometry-based assays to look at glucose uptake. So 2-MBDG is a fluorescent glucose analog, and then MitoTracker's red and green to look at mitochondrial membrane potential. And so as these are flow cytometry based assays, they are not real-time bioenergetic assays, but they can give us kind of a surrogate measure of what may be going on in terms of glycolysis versus oxidative phosphorylation. And so when we looked at glucose uptake in NOD B cells compared to non-autoimmune B cells, from a mouse that will not develop diabetes, what we see is that at baseline, glucose uptake is really the same. However, with activation, glucose uptake increases for both the non-autoimmune Black 6 mice B cells and the NOD B cells. However, the increase in the NOD B cells is significantly higher than what we see in the non-autoimmune prone Black 6 B cells. And so we see exaggerated glucose uptake in NOD B cells. We really didn't see much change in mitochondrial membrane potential. And so we know from the initial paper that showed HIF1 alpha was important in other autoimmune disease models that HIF1 alpha really drove a glycolytic expression, or drove glycolysis to facilitate that expansion. And so it looked like we have potentially lots of glycolysis. That doesn't seem to be the issue. And so we wanted to look next to see what is the expression. What is HIF1 alpha expression doing in these activated B cells as well? And so we activated B cells using 3 different ways to kind of come at the activation from 3 different angles and different signaling pathways. And what we saw fairly consistently was that HIF1 alpha expression was lower in the NOD or the diabetes-prone B cells compared to that of the non-autoimmune B cells. And so specifically, when we simulate receptor signaling through the B cell receptor using anti-CD40, we see a significant difference at 24 hours in the HIF-1 alpha expression in NOD versus black 6 mice. And so that's what you see on the far left of your graph. And so it looks like there may be something that is different about how or whether HIF-1 alpha controls glycolysis or how it mediates glycolytic pathways to promote expansion of regulatory B cells. And so we wanted to look more specifically to get an idea of what it could be doing at the gene expression in NOD and black 6 mice after exposure to hypoxia. And so we know that HIF-1 alpha is induced in the settings of hypoxia. And so by culturing overnight in a hypoxic environment, we are forcing HIF-1 alpha expression. And when we force HIF-1 alpha expression in both populations of B cells, and we look at HIF-targeted glycolytic gene expression, we see that compared to the black 6 mouse, those glycolytic genes are less expressed in the NOD compared to the black 6. And these are all genes that were found in that initial paper to be expressed less with HIF-1 alpha deficient mice. And so this looks like some sort of defect with how HIF may be driving that response. But at the same time, we know that ultimately we want to know what is it doing to those regulatory B cells, because our ultimate goal will be to skew the repertoire towards those protective rather than autoaggressive B cells. And so we also looked at differential gene expression of 19 common marker genes and transcription factors that were common throughout tissues for regulatory B cells. As we learn more and more about regulatory B cells, we are learning more and more about how to define them, but there's not a universal transcription factor at this point that we can say this identifies a regulatory B cell. And so to make sure we're encompassing genes that would be common anywhere, we looked at these 19 genes. And we looked at NOD B cells in normoxia, so without driving HIF activation, and in hypoxia, so after incubation with hypoxia. So we're driving HIF activation. What happens to those regulatory B cells? And what we see is in hypoxia, they are not increased, they're actually decreased. And so even when we are sufficiently driving HIF-1 alpha, we don't see an increase in those regulatory B cell genes. This is just broadly, obviously there are many more mechanistic studies that we will need to do to parse this out to look specifically, but overall, hypoxia is not driving differential expression of the common regulatory B cell markers. And so it led us to this thought that potentially HIF-1 alpha expression in the NOD mouse specifically is insufficient to drive that regulatory B cell expansion. And so if you think about your effector response, and in individuals with autoimmune disease, you have antigen counter, environmental cues, and as HIF-1 alpha increases in both autoimmune and non-autoimmune disease, HIF-1 alpha then starts to drive the suppressive response. HIF-1 alpha expression will start to drive expansion of regulatory B cells to tamp that reaction down. But there's something about HIF-1 alpha and how it makes it unable to drive that similarly in individuals with autoimmune disease. And so it seems that this data could be consistent with historical data from the NOD mouse. So this is a study that just looked at response to immunization in the NOD compared to other black 6, BALB/c lines. And you see in the NOD mouse, you see a very exaggerated response to immunization that eventually comes down. And so that indicates an inability to effectively suppress the response when it's appropriate. And so it seems like this data could be consistent with what has been seen historically in NOD mice. And at the same time, we see in children with type 1 diabetes, decreased subsets of regulatory B cells have been identified in the blood. And so is there something about this response that is preventing these B cells from making IL-10 and really controlling the response to antigen that you see in people without autoimmune disease. And so overall, this is all really exciting and for us, but my lab's mission is to use basic and translational research to understand the type 1 diabetes pathogenesis and identify therapeutic targets. And this obviously is very, very early stages of work. As part of the Diabetes Center, I get to work closely with our clinical colleagues and other colleagues doing translational research. And so in addition to this work, I am also able to take care of patients who have those early stages of type 1 diabetes. And we were able to treat the first patient in Indiana with an antibody called teplizumab or anti-CD3, which was shown to delay the onset of type 1 diabetes. And so it was really exciting because teplizumab is something that started as basic science work, started on the bench back in 1993. And now we are able to treat and continue to look for additional targets for other options to prevent development or delay development of type 1 diabetes. And so I feel like my work and specifically within the Diabetes Center has really come full circle and has allowed me to look more specifically at these basic concepts, but also apply them and be able to use them in clinical practice. So that was a brief whirlwind, quick overview. I have lots of people to acknowledge. My current lab, Jesse Bernard, is a postdoc here and mentors both at Indiana University and at Vanderbilt, and also my funding. Thanks.